Good day, we are Group 3 and we will be discussing Marine Biomaterials and Bioengineering. This study is entitled Naturally Prefabricated Marine Biomaterials, Isolation and Application of Flat Chitinous 3D Scaffolds from Iantella labyrinthus. First up, our objectives. It generally aims to isolate based scaffolds from Iantella sponges, and it specifically aims to answer the following. First, give a background information regarding chitin-based scaffolds. Second, discuss the morphology and importance of marine sponges, primarily Iantella labyrinthus. Third, what marine biomaterials can be isolated from this species and the relevance of it. Fourth, give an overview of this sponge species as a potential source of natural compounds for pharmaceutical industry, tissue engineering, and regenerative medicine. Fifth, compare the performance of microtubular organization of I. labyrinthus skeleton for new generation hemostats to cellulose-based. Sixth, discuss the methods of stereomicroscopy, imaging, immunostaining, and fluorescence microscopy. Next slide, please. And now for our introduction, we all know that the marine environment covers more than 2/3 of our planet. Yet, it remains largely unexplored and requires further research. It harbors a wide variety of molecularly diversified range of organisms when compared to terrestrial environments. Despite this, marine environments are still promising source of commercially viable biotechnological products and biomaterials that are essential in the treatment of disease and has been used as a template to develop therapeutic drugs. And in particular, marine sponges, a well-known diversified multicellular invertebrates found in abundance in marine benthic environments, are acknowledged as one of the most prolific known sources of pharmaceutically significant biologically active secondary metabolites. Researchers have been studying marine sponges for biomaterials as well as pharmacological possibilities, as sponges are major contributors of biomaterials and natural products for biomedicine according to a study conducted by Bhatnagar on year 2016. Then, the intention of this study was focused for the first time on the marine sponge Iantella labyrinthus as a novel potential source of naturally pre-structured bandage-like 3D scaffolds which can be isolated simultaneously with biologically active bromotyrosine. Furthermore, for the first time, we also choose human-induced pluripotent stem cells or iPSC-CMS to investigate the suitability of the scaffold for cell culture made of Iantella labyrinthus chitin. Demos sponges' ability to regenerate tissues and skeletons, as well as flourish under marine farming settings or in biotechnological sustainable biomass production based on cell culture is a crucial feature considered upon the selection of these organisms. And as a result, a current trend in demo sponge application is centered on the development of methods for extracting both secondary metabolites and biomaterials. Thus, researchers believe that these sponges could be used as a ready-to-use structures to replace damaged skin fragments, or as wound dressing alternatives, or for plastic surgery treatments. Next slide, please.
For the materials and methods, location and collection. Sponge Iantella labyrinthus samples were collected in Western Australia in March 2015 at a depth of 35.3 to 35.5 meter. Samples were collected either by scuba diving, dredging, or hand-picking in shallow water on the rocks. These were photographed and directly preserved in 99% ethanol. For the isolation of chitinous skeleton from sponge and identification of selected bromotyrosine, sponge skeleton will be washed by distilled water 3 times for removal of water-soluble compounds. By the use of methanol, bromotyrosine is extracted. Residual fragments were placed into plastic boxes with 2.5 meter NaOH at 37 degrees Celsius for 72 hours to remove cells, proteins, and pigments. To remove residual calcium carbonate, samples were treated with 20% acetic acid at 37% for 5 to 8 hours and washed with distilled water. This isolation procedure was repeated 3 times to obtain colorless tubular scaffolds. The Shiamatsu HPLC system is used to analyze methanolic extracts of sponge fragments. This was accompanied by a UV-vis detector. For compound separation, a solvent system containing MECN and H2O was utilized. For stereo microscopy imaging, photographic images were taken with a Case VHX 6000 digital optical microscope. For the differentiation and culture of human iPSC CMS, the sponge chitinous scaffolds were prepared and autoclaved in PBS. Sterile scaffolds were cut in and pre-incubated either with the extracellular matrix, mimetic Geltrex, or DNEM. To obtain cardiomyocytes from iPSCs, cells were differentiated using modulators. After differentiation, iPSCs CMS were maintained in standard cardiomyocyte medium with 2% B27 supplement at 37 degrees Celsius, 5% CO2. After a period of 1 month maturation, iPSC CMS were detached using collagenase B for 30 to 60 minutes at 37 degrees Celsius. iPSC CMS on chitin scaffolds were cultured for 20 days, with medium exchanged every 2 to 3 days, and documentation was performed using light microscopy. So that's all for the methods.
And for the procedures, the samples collected are the Ientella labyrinthus, wherein, in order to establish the tissues from these mechanically rigid and dark reddish pigmented meshwork sponges, 2 separate sponges will be inserted and washed in a distilled water for 3 times, or would undergo treatments of 2.5 mm and AOH solution at 37 degrees Celsius. Also, both kinds of extracts will contain bromotyrosines through the use of methanol. After the 24 hours insertion of Ientella labyrinthus in the distilled water at room temperature, its typical flat, mesh-like architecture should be visible. Meanwhile, after the 48 hours treatment with 2.5 mm and AOH at 37 degrees Celsius, bromotyrosine containing extract and bromotyrosine containing flexible skeleton of Ientella labyrinthus should also be seen. There are alternating treatments that can be used to obtain translucent, elastic, flat, and scaffolds from the cell-free skeleton of Ientella labyrinthus. This would be successfully isolated if there's an alternating use of acid and alkali treatments for 72 hours at 37 degrees Celsius. Therefore, it would produce a ready-to-use flat 3D chitinous scaffold isolated from Ientella labyrinthus remains porous after those analytical procedures. The Ientella labyrinthus had been investigated more with respect to their application for growth of human iPSC CMS, or the human-induced pluripotent stem cells. In that case, this would be followed by the cultivation of iPSC CMS and will be investigated on pure chitin scaffold, then compared to material that was pre-coated with the extracellular matrix mimetic Geltrex, which is applied in standard iPSC CM culture approaches using commercial plastic plates. For the schematic image of sponge structure, it needs to be cultured in transfer plates with low-medium levels. 8-week-old iPSC CMS must be supplied by medium absorbed by the sponge structure and cultured in an uncoated and Geltrex-coated sponge scaffolds for 20 days. Generally, there are 2 experiments that will be performed using the iPSC-CMS from different healthy donors. This is the iPSC-CMS on Geltrex-coated versus uncoated chitin scaffold of Ientella labyrinthus. Furthermore, there's a staining of A-actinine, Ki67, and cell nuclei to document the cell alignment and proliferation of iPSC-CMS on Ientella labyrinthus scaffolds. Additionally, there's relative quantification to be performed through cell counting, which is based on different images from 2 independent experiments. Lastly, there will be observation and documentation of iPSC-CM attachment using fluorescent microscopy. Thank you.
Results and discussion. Isolation of 3D chitin scaffolds. From the cell-free skeleton of I. labyrinthus, transparent elastic flat scaffolds can be made. These one-of-a-kind natural gauze fabrics constructs can be modified and take on the shape of the hard surfaces they placed on. The ready-to-use flat 3D chitin scaffold isolated from I. labyrinthus is porous and mechanically strong enough to be manipulated as shown in this image. These characteristics, together with the capacity to expand with a range of liquid in a matter of seconds due to capillary effect, and structural stability after sterilization up to 200 degrees Celsius may be enabled for practical use of such scaffold in regenerative medicine, employing tissue engineering principles. iLabyrinthus provides a ready-to-use isolated flat 3D chitin scaffolds that preserves its porosity nature while being mechanically robust enough to be manipulated manually. 3D cutaneous scaffolds isolated from iLabyrinthus is a microtubular interconnected meshwork that can absorb water and serve as a medium for cell culture due to capillary pressure. The permeable region allows air to travel through. 3D scaffold, single picture were stacked in cardiomyocytes. A-actinine is a structural maker for detecting sarcomere units. The repeatable consistent orientation of the sarcomere in iPSC-CMS attached to the chitin scaffold clearly shows that iPSC-CMS align parallel to the long axis of the chitin scaffold. In this regard, it's vital to note that stretching pressures have been identified as key stimuli for iPSC CMS structural and functional maturation, such as engineering muscles. The creator protein, Ki67, was created to assess the proliferation of iPS CMS on the sponge scaffold, which has been utilized to measure CM proliferation in various studies. On Geltrex-coated scaffolds, positive cell walls were found to be 4.7 positive negative 0.7% and 7.1 positive negative 1.8% on pure culture uncoated scaffolds. Furthermore, studies on specific location is identified. C position, represented by red lines in YZ projection, demonstrate the attachment of iPSC CMS to the upper surface side areas and upper bottom of the chitin fibers as shown in the figure. 3D scaffold, single picture were stacked in cardiomyocytes. A-actinine is a structural maker for detecting sarcomere units. The repeatable consistent orientation of the sarcomere in iPSC-CMS attached to the chitin scaffold clearly shows that iPSC-CMS align parallel to the long axis of the chitin scaffold. In this regard, it's vital to note that stretching pressures have been identified as key stimuli for iPSC CMS structural and functional maturation, such as engineering muscles. The creator protein, Ki67, was created to assess the proliferation of iPS CMS on the sponge scaffold, which has been utilized to measure CM proliferation in various studies. On Geltrex-coated scaffolds, positive cell walls were found to be 4.7 positive negative 0.7% and 7.1 positive negative 1.8% on pure culture uncoated scaffolds. Furthermore, studies on specific location is identified. C position, represented by red lines in YZ projection, demonstrate the attachment of iPSC CMS to the upper surface side areas and upper bottom of the chitin fibers as shown in the figure.
The pictures shown are the uncoated and Geltrex-coated sponge. Fluorescence microscopy was used to document induced pluripotent stem cell-derived cardiomyocytes or the iPSCM adhesion. The maker protein Ki67, which has been used to assess CM proliferation in several investigations, was developed to investigate the proliferation of the iPSCCMs on the sponge scaffold. Positive cell walls were found to be 4.7, positive negatively 0.7% on the Geltrex-coated scaffolds and 7.1, positively negatively 1.8% on pure culture uncoated scaffolds. In addition, the attachment of iPSCCM to the upper surface, side areas, and upper bottom of the chitin fibers are demonstrated by analysis of particular region in defined Z position indicated by red lines in YZ projection. Next slide. 3D chitin scaffolds of Poriferan origin as a alternative gas fabric. Iantella labyrinthus chitinous scaffold quickly absorbs wine blood off the skin surface. In contrast to a previous experiment that used synthetic gas fabric, sponge chitin is able to absorb blood quickly and deeply into microtubes due to its capillary effect.
In this study, it proposes that the use of a naturally pre-structured fibular chitin scaffold of Poriferan origin can be a crucial step in determining better hemostatic dressings. Additional, future research should focus on this topic to assess the many outcomes that may emerge from their usage, notably in terms of positive, negative consequences, and safety. Furthermore, in order to develop unique and effective hemostats, the biological performance of sponge chitin-based absorbable hemostats must be evaluated and compared to the reports for cellulose-based materials for safer hemostatic, primarily in places where access to present existing hemostatics is limited agents.
Significance of this research have found out that chitin of Poriferan origin has been recently recognized as a renewable source of uniquely, naturally porphyricated 3D construct. Moreover, diversity in size, shape, and porosity of such chitin is based on structural peculiarities of the original sponge skeleton. Aside from that, this research also determines that lanthelids can obtain good growth under marine farming condition due to special ability to regenerate the chitin-based skeletal structure very quickly. In addition, it has been proven that chitinous scaffold or Poriferan origin possesses tubular structure and remarkable swelling properties due to capillary forces and the porosity of this material, which evolved due to millions of years and eventually optimized to support cell growth and proliferation. Lastly, this research found out that labyrinthus has remarkable biomimetic potentialities as a source of natural compound useful for pharmaceutical industry and of new biomaterial of tissue engineering and regenerative medicine simultaneously.
And here are our references. Thank you for listening.